All right, everybody. Welcome to another interview. I'm really excited to interview Sue Ishaq. Actually, I'm not sure if I'm pronouncing your name. Did I get that right?
I say Ishaq because it's a little bit more phonetic, but it's a name that I picked up from an ex-husband, so I actually don't care how it's
Okay.
pronounced.
All right.
Makes it easy.
All right. So
That's another story. Yeah,
we're interviewing Sue today. You're from the University of Maine, or am I getting that wrong? Is
I
Southern?
am now. Yeah. From is kind of a loaded word because this is the fourth university that I've been at.
All right. Well, we'll get
Yeah,
to that. Actually, let's
yeah.
get off there. So why don't you, if you don't mind, just taking us through the steps of your career to get to your fourth university that you've been at.
Yeah, it's been interesting. So in sixth grade, I decided that I was going to be a veterinarian, an animal doctor. That was all it was going to be. I wouldn't take no for an answer. And then the first 3 weeks of my undergraduate in a pre-veterinary program, because you have to get a bachelor's degree and then go to the vet school in the US, I met all the other pre-vet students, and they were so much more driven and competitive because there's only so many spots to get into vet school. And I was like, no, I want something more laid back. So then I decided that I would go to graduate school,
Right.
right?
That's more.
Joke was on me. Yeah. Yeah. So I, for 4 years, wasn't really sure what I wanted to do for graduate school, but something with animals and wild animals and conservation. So I just took a bunch of different classes. My degree was really flexible in that way. So anything from ocean ecology to wildlife ecology to animal behavior. And that was really helpful because I didn't know what I wanted, and I didn't know what being a veterinarian was like until I got more familiar with it. So I figured science was sort of the same. So after my bachelor's, ironically, I then got a job for a year at a veterinary hospital doing tech work.
Okay.
Confirmed that I didn't want to be a veterinarian, ended up getting stuck there for 3 years because it coincided with the 2008 crash. And
Yeah.
all of a sudden, people just didn't have money to take on graduate students. So I sort of bided my time. And after 3 years, I thought I should go back and actually get a research related job because my skills are decaying. And the older I get, the less helpful I am, and nobody wants to take me on, right? So at a holiday party at that veterinary hospital, I got into a conversation with the spouse of a veterinarian who had just started working at the University of Vermont
Okay.
and said, "Hey, we have a new department chair. They're renovating the farm. I know they're hiring. Like, you should go talk to Andre Wright and see if he's got any jobs for you for research." So I emailed this department chair kind of out of the blue. I said, "I'm looking for research jobs. Do you have anything?" And he said, "Well, I don't have any research jobs, but have you ever considered going to graduate school?"
Yeah.
Like, have I? Yeah. So we met for coffee a couple times and discussed my background and what sort of project that would be. And when I was an undergrad, I specifically avoided microbiology, genetics, and computer software. And then it turned out that my project was going to be microbiology, genetics, and computer software. The good news is that they weren't anything like I thought they were going to be. And so when I actually took those classes as a graduate student, I really loved it. So I started my graduate work in 2010 to 2015, cataloging the microbes that were in the rumen and other parts of the gut of the North American moose, which
Cool.
hadn't been done, yeah, since before I was born,
Right.
and clearly hadn't been done with sequencing tools. So we started that turned into me doing a whole lot of anaerobic culturing on some of those samples using a culturing chamber, and coming up with a few isolates that we turned into like a probiotic, like a
Okay.
moose probiotic that we then gave to lambs to see how the lambs would grow with like marginal success.
Okay.
um, it certainly didn't do any worse. So that was like the end of my PhD. And um, I wanted to continue in animal science, but I was looking to branch out a little bit. And the second half of my PhD ended up being really data driven and data analysis heavy in terms of DNA sequencing analysis. So the job I ended up getting was a postdoc out at Montana State University in Bozeman, Montana, with Carl Yeoman, that was mostly going to be doing data analysis, primarily on rumen related microbial data sets, but it branched out into soil and water and other things.
Okay.
That postdoc was supposed to be 2 years, and 6 months in, stuff happened, and we started running out of research money sooner than expected.
Right.
So I scrambled looking around for another postdoc position because I didn't want to move across the country again quite so soon. I managed to find a postdoc position in a lab that I had collaborated with as a data analyst. And so I moved across the street into a weed and wheat ecology lab, which
Okay.
was totally new for me.
Yeah.
Fabian Manalid, and I started learning about soil bacterial communities for a year. That money also ran out sort of sooner than I would have expected, so I ended up jumping ship a little bit early. I went to the University of Oregon in Eugene
Okay.
for about a year and a half, which also ended up earlier than expected. Finances get you every time. But I moved out to Oregon to be part of the biology and the built environment center, where I would research microbes indoors and primarily what people are putting down and what we're picking back up and how they kind of move through spaces.
Okay.
That was a really cool position because there were a bunch of research scientists and there were or biological research scientists, and there were a bunch of research scientists who were architects. And so we mashed those two things together to understand how people use buildings and how microbes move through there. But it wasn't, I mean, I started in animal science. I really wanted to get back to animal science. So I was just sort of waiting for this position to open up. And a friend actually sent it to me. I hadn't even seen the call, and applied, got an interview, came out here to talk, negotiated a new position, and then moved back across the country all in about 30 days' time.
Wow.
Yep. I don't recommend it, but I didn't, yeah, kind of how it
No,
rolled
no
out.
stress with that, I'm sure. Right.
Say that again?
No stress with that at all, I'm sure.
Oh gosh, yeah, yeah. Talk about an adrenaline rush trying to manage the logistics of that. Luckily, I had very supportive departments and family and a partner, and so that all made it extremely easy for me to kind of cut ties and dramatically move across the country. So yeah, so I've been at the University of Maine since September 2019, and I was only here for what 4 or 5 months before all of a sudden we went into semi-pandemic lockdown. So it's been a really interesting first year and a half for me.
So you don't have a normal, like you don't know what normal is, right? So when we say, "Oh, we're gonna
No.
get back to normal," you're like, "What? I don't know what that is." But
Yeah, and I also the first year I was here, I was sharing space with a faculty that was leaving, and so the space and all of the components in it weren't really designed for microbiology. It was sort of shifting. So it took quite a bit of time to catalog everything, move everything around, figure out what I wanted to keep, buy new stuff, kind of transition between their work and mine. So it's been, I feel like, one hurdle after another, but
okay,
everybody in the department has been really supportive and really helpful. So it's kind of eased that transitional stress.
that's I think one of the most important things. If you have a friendly department that you're moving into, even if you're not a perfect fit, that's a much better fit than the ideal space with people that aren't going to help you out very much.
Absolutely. And I really liked Maine. I mean, it seems like a smaller school, and a lot of people felt like it was a step down when I went from the University of Oregon to the University of Maine. But I said, actually, this is an agricultural based school. The department is set up specifically for what I want to do. And
Right.
so, yeah, it's a smaller school, fewer students, fewer resources, but it's more applicable to me. So it's actually much more productive and a much better fit for me.
Excellent.
Yeah.
If you wouldn't mind, maybe like 2 minutes. Like what do you do? What's your research focus? And keep in mind, we're a 100 level intro bio majors class. So,
Yeah. So what I am back in after such a long hiatus through my career is into gut microbiology,
okay.
specifically of animals and mostly focusing on livestock. But I kind of have to go where the money is, and there's a lot of interesting questions to be had in wild animals specifically or humans specifically. So most of what I work on is the bacterial community as a whole and how that changes under different circumstances and how that can help or hurt the host. So I don't
Okay.
know if you've gotten into host associated
So
microbiology yet.
no, not really. So we in our lecture, I mean, I had a bacteria lecture today, so that was basically an entire one day for an entire domain. And then I'm sure that they'll come up with a whole bunch in other areas. But I mentioned as part of that, like, oh yeah, we have a microbiome, and this can influence our health. There's some data that suggests that. Would you be able to kind of put that into a more, I don't know, elegant phrase, or maybe not elegant, but a more up-to-date phrasing than I, a non-biologist or non-microbiologist, can do?
Um, yes, elegant is definitely not the right description because I usually respond with humans are really gross.
Okay.
So all over all of our surfaces, including the internal surfaces, but not the internal tissues necessarily, we are just coated with microbes. And on our skin, they come and go. In our hair and on our eyes, they kind of come and go. The ones that are in your gastrointestinal tract are really interesting. So you are ingesting microbes all the time, either from the air or every time you put something in your mouth or from your coffee mug. Anytime you eat food, it's got microbes in it. And you will select certain microbes to continue living in your gut based on your internal circumstances. So, like the pH in the different parts of your digestive tract, the amount of salinity that you've got going on or in your diet, the type of foods that you eat. So, if you eat, a lot of chocolate chip cookies, you're going to recruit microbes and feed microbes that particularly like breaking down those simple sugars. Whereas if you eat a lot of fibrous vegetables like you're supposed to, regrettably, I have to tell you, you should be eating fiber and not chocolate chip cookies.
Burn.
Yeah, like I got into gut microbiology to prove the opposite, but here we are. You really need your fiber. Part of that is because plant fibers are really big, complex uh, chemical structures, and you can't break down most of those all on your own. You actually need microbes to do it for you.
Right.
And different microbes have different capacities in terms of the enzymes they have and what they like to eat. So they'll break down those complex large fibers into small little components that other microbes can eat. They themselves, or they then eat. They can create byproducts that other microbes will eat as kind of an accessory. There's
Right.
stuff that you can absorb. And so you end up creating this like really thriving community with lots of different members that are all doing something different. And so they produce some things that are really helpful for you. They'll produce things that will actually kill off other microbes, or they can, occasionally make you sick. So there are a lot of blank spots about, what happens when we do this or what happens under these conditions. So my work kind of focuses on like, all right, now we're sick. We've got a disrupted community. We need to set it back to what it was before.
Right.
How can we best do that?
And you're doing that largely in the context of like agricultural animals, like you mentioned moose as part of your training, but I would imagine like cows and sheep and other vertebrates for the most part.
Yeah, my position is specifically to support Maine agriculture. And so that's mostly what pays the bills.
Okay.
And you get a really interesting animal model with livestock because, yeah, they're out kind of doing their own thing, but you can keep them under very specific conditions. And
Right.
we've also been breeding them for a really long time. And because we've been breeding cows to produce milk, not necessarily to be, good community members with their microbes, it started to change the cow growth cycle, and it started to change the cow immune system a little bit in the way that we produce them, that changes the way they interact with their microbes.
Oh, yeah, I would not have thought about that. That's really interesting.
Right? Yeah. So if you think about like a wild cow, right, or a wild sheep, you can compare your wild and your captive populations and then your also domesticated populations and kind of understand how we might disentangle the role of the microbe versus the role of the host and what's
Yeah,
situational and yeah, what we
it's an
kind of recruit
excellent
over
system
time.
to try to understand that. I mean, I've been hearing about microbiomes for a long time, but it's always kind of just like sequencing data. Here's what's there, and not
Yeah.
a lot of like, here's what it means or why it's relevant, right? There's some work that I'm not all that familiar with of like doing transplant, like fecal transplant kind of stuff to try to, and then suggesting like, oh, that changes like brain chemistry or something, some weird stuff that I kind of suspect is wrong, but is
Yeah, yeah, certainly the things that you can show in a mouse model are different than what really ends up going into humans. So if you look at like pop science or what's in the news, you'll see like those glorious images of, the Michelangelo done in microbes kind of thing. And it's a little less glorious and a little bit less well understood than that would make you think.
Right.
And of course, like mice have different digestive tract setups, and they've got different placental structures that will change how they interact with microbes and how their immune system develops. So like you can't directly go from a mouse to a human because our interactions are different. So there's so much that you can do with microbiomes, and it's a lot of it is sequencing data, but a lot of it is also anaerobic culturing or trying to look at that whole community using kind of high-tech microscopy and understand like, okay, who likes to be out here and who wants to be down here on the surface next to the host, that sort of thing.
So if one of my students is interested in microbiology, basically there's a lot of work to be done. Yeah.
Absolutely. And there's so many different ways that you can come at this. I mean, I worked in like 4 different fields of microbiology that all sort of ended up being kind of knit together because now I've got the tools that are kind of inherent to the slightly different facets of science.
Cool. One more question, and then I want to play a quick game, if that's all right. So the last question is, what's the most interesting thing that you've discovered, or the thing that you're most proud of, or if you had, or your best paper, I don't know, however you want to interpret that. What are you most happy with about the work that you've done?
Wow, that's a tough call. I've got 2. So the most helpful work I think I've ever contributed was a project that looked at the calf digestive tract in 9 different locations along the digestive tract, and it looked at the digesta, so like what was in the middle of your intestines versus what was scraped off on the side.
Okay.
And in each of those different anatomical locations on your GI tract, there were different communities based on the local circumstances, but also what was in the digesta was different from what you find on the side. So there are certain microbes that like to hang out and attach to your epithelia, and they will create biofilms, they'll latch on, and they will interact with you in different ways than the ones that are kind of passing through in that digesta.
Okay, yeah, that makes sense.
Yeah. So like being able to, and I didn't come up with that project. I was part of it as a postdoc. So there's like a lot of people involved with that project. So I think showing that anatomical specificity and spatial location in the gut is really important, because if you want to develop a therapeutic, you need to be able to target it where that microbe is going to live, right? But my personal favorite tends to be some of the protozoal work that I did as a postdoc or as a PhD student and hope to revisit, just because like rumen protozoa are so cute and they're so fun, and like nobody ever wants to
We're getting
study
into
them.
protists next week in class. They're definitely one of my
Yay.
They're really fun. There's again so much there to go. And it's an intro class. I could spend a semester on like each of the lectures that I'm having for the first part of the class. But yeah, protozoans are awesome.
Awesome.
Cool. All right, so I have a game. If you are up for this, I'm going to share a image that I showed in class, and I kind of stripped it down so it's just the image, and you can interpret it and then try to explain it. And if it fails spectacularly, don't worry, I can just cut this out, and that'll be the end of it. So,
Okay.
all right, so can you see that?
Yes. Oh, okay. I actually do sort of know this one. I
Okay.
can't explain it with any specificity. It's been a very long time since I've taken microbial cell biology. Okay. So, microbes are really cool in that the flagella setup that they've got are different between different taxa. And so the way that they create proteins to actually shape that machinery is a little bit different and helps you discern between like bacteria and archaea, or bacteria and fungi, or I guess like giardia if you're going for protists. Yeah. So beyond that, I don't remember any of the specifics. So hopefully that's not on the test.
I think all that I used this slide for was just to show what a flagella is, or flagellum is. I just made the point that it's anchored in usually a double membrane, or at least in this case in a double membrane in bacteria. And And then I basically just said that they're really cool and complicated, and there's this fascinating evolutionary story about how, you know, flagella came to be. That's, you know, there's a subfield of a subfield of a subfield of microbiology that studies flagella, trying to figure out how they work and how they evolved and all of that stuff. So
Yeah, thinking back many years ago now to my freshman biology, it just sort of seemed like, you know, you had a flagella and it does this and then you're good to go.
right.
And it wasn't until I ended up getting into more of the graduate work where I realized that there's so many differences, because microbes are most of the tree of life. They've been here for so much longer than anything walking around. And so they've got like really complicated and interesting, yeah. I like microbes because you can never really assume what's going to happen or what you're going to find. You have to actually go and find out.
And see if you're correct, or most likely you're wrong. Cool. All right. Well, thank you very much. I hope the students enjoyed this. It was a fun conversation. Hopefully you had a bit of fun.
Yeah. And they are welcome to contact me or follow me all over social media at Dr. Sue Ishaq.
Okay.
If you want to know more about how gross humans are.
I'm trying to figure out where the best. I'll put it into the next lecture that I have after they see this video, so that they can look that up if they want to. If not, they can Google your name. I think you're probably easy enough to find that.
I'm not the only S Ishaq, but I am the only Sue Ishaq out there.
Yeah.
So got that cornered.